So thank you, Ronan, for the introduction, and thanks to the organisers for the kind invitation to present. So today I'm going to walk you through some of the most recent advances in the biology of a key player in neurodegenerative disease, namely tau. So it actually follows on very nicely from Irasima's focus on beta amyloid and amyloid therapies. Um, Essentially, I'll discuss some of the, I'll move from the basic structure of tau all the way through to current knowledge on biomarkers, tau biomarkers, and tau therapies. So let me start with a very simple question. Why is it that two patients who can both have Alzheimer's pathology progress at completely different rates with different symptoms and have different brain regions affected? Well, for a long time, we didn't have a very good answer to this. We thought of tau protein as being one species which behaved more or less the same way in every disease and in every case. But over the last few years, that view has completely changed. And we now understand that the tau protein exists in multiple distinct forms with very different structures, different modifications, and different biological behaviors across different diseases. Okay, so I'm just going to go back to the start, focus on what exactly is tau. So tau is a protein which is encoded by the MAPT gene on chromosome 17, and it contains 16 exons, and through alternative splicing, it results in 6 protein isoforms. So these differ due to the number of N-terminal repeats, so N1 or 2, which can have 0, 1, or 2, and also due to the number of microtubule binding repeats, which are here. So they can either have 3 of these or 4, as you can see in some tauopathies. So this distinction is important because different tauopathies are associated with different isoform compositions. So mutations in the MAPT gene can also act in several ways. Some affect splicing, particularly around exon 10 here. And this shifts the balance between 3R and 4R tau, while others occur within coding regions, especially within the microtubule binding repeats. And these mutations directly influence tau structure and aggregation. So in addition to all of this, tau can be heavily post-translationally modified, and the most prominent and probably well-known, which you're familiar with, is tau phosphorylation, but it can also undergo acetylation, ubiquitination, and glycosylation. So you need to remember that a lot of these modifications occur naturally and have physiological importance in the normal cell to maintain healthy cell biology. But in disease, they can contribute to, let's say, tau misfolding and aggregation. So this slide captures the core transformation of tau during disease. Normally in healthy cells, tau binds and stabilizes microtubules, and it's predominantly located in the neuronal axon. It's dynamic, it's soluble, and structurally it's intrinsically disordered, so it doesn't really contain much structure. So it primarily controls and is regulated by phosphorylation, and it's, as I said, it supports axonal transport and neuronal integrity and communication. However, in disease, tau detaches from microtubules. It mislocalizes from the axon to the somatodendritic compartment of the cell, and it can undergo abnormal PTMs and post-translation modifications. So it can be hyperphosphorylated, and it also can undergo truncations, and these change the basic biochemistry of the protein itself. So as a result of all of these disease processing, let's say, tau can form soluble oligomers, and these ultimately lead to insoluble fibrils and neurofibrillary tangles in disease. So importantly, these pathological forms of tau can also spread between cells in a prion-like way, and thus these propagate the disease itself. So the key idea is that it's not just aggregation, it's a state transition from a functional protein to a misfolded self-propagating system. So tauopathies are a family of progressive neurodegenerative disorders, and these are characterized by abnormal accumulation of tau in the brain. and these leads to cognitive, behavioral, and memory impairments. So there's just over 25 distinct tauopathies identified, and these can be broadly classified into primary tauopathies, which where tau dysfunction is the primary driver of disease. And these include diseases such as Pick's disease, progressive supranuclear palsy or PSP, and corticobasal degeneration or CBD, but also secondary tauopathies, such as Alzheimer's disease, where tau aggregation occurs alongside other pathological processes, such as beta amyloid. Also, the tauopathies are classified according to the different isoforms. So those diseases which are enriched in 3R tau include Pick's disease and familial frontotemporal dementia, whereas 4R tauopathies are enriched for, as I said, 4R isoforms of tau. And these include PSP, corticobasal degeneration. And then you have diseases such as Alzheimer's disease, which is actually a mix of 3 and 4 tau. So now I'm just going to walk you through some of the most recent research highlights in tau biology. One of the kind of most recent advances, as I said, for a long time, tau has been shown to be intrinsically disordered and not much has been known on its structure. However, recent structural studies have shown that tau is not a single uniform aggregate, but it's rather a family of disease-specific filament folds. So cryo-EM work first established that tau filaments in Alzheimer's disease have a characteristic architecture, and later studies have shown that PSP, corticobasal degeneration, and Pick's disease, for example, also have distinct core folds of the tau protein. And this supports a strain-like conformational model of tauopathy. This diversity also appears in familial MAPT mutations. And so, for example, in the P301 and P301T, LNT mutant species of tauopathy, cryo-EM has also revealed novel tau folds in these that differ from one another and from, let's say, the folds which are observed in sporadic forms of the disease, showing that even a single gene mutation can produce multiple pathogenic conformations. So tau exists as a diverse set of proteoforms rather than a single uniform protein. And this is shaped by, as I said, alternative splicing, post-translational modifications, and also proteolytic processing. So recent large-scale analyses of human brain tissue have shown that tau diversity is far greater than previously appreciated, with extensive variation in the modification patterns and cleavage sites across the different tauopathies. So these molecular differences are not random, but rather follow disease-specific patterns that correlate with pathology and progression. So importantly, different tauopathies can be distinguished based on their unique molecular signatures. And this suggests that tau biology varies significantly between diseases rather than following a single common molecular pathway. And these support a model in which tau pathology is driven by specific combinations of molecular changes, be it truncations, PTMs, etc. And these can influence how tau aggregates, spreads, and also contributes to neurodegeneration. I think I zoomed in there, so. So tau spreading is now understood as a regulated multi-step process rather than being a simple passive release into a cell. So it involves tau being released from cells, taken up by neighboring neurons, trafficked intercellularly, and ultimately seeding further aggregation. So recent work in our institute and others has highlighted the role of extracellular vesicles in this process. So tau filaments, which have been observed in EVs, have been shown to be tethered to the membrane, and this suggests that tau spreading is actually selectively packaged rather than randomly released into the extracellular environment. So once outside the cell, tau is taken up through endocytic pathways, And these include mechanisms involving HSPGs, so heparin sulfate proteoglycans. And after internalization, tau enters the endolysosomal system, which acts as a key decision point where tau can either be degraded or go on to seed aggregation. Importantly, newer studies suggest that tau seeding can occur within these compartments, even without full membrane rupture, making intracellular trafficking pathways central to disease progression. Tau can also undergo liquid phase separation, and these form condensates which are normally dynamic and quite reversible. However, in disease and pathological conditions, these condensates can act as nucleation points and sites for aggregation, and these obviously may promote the formation of insoluble tau fibrils. So this positions phase separation as an early step linking soluble tau to aggregated tau species. These condensates are not isolated. They interact with cellular quality control systems such as chaperones and degradation pathways. And these also may influence whether tau is cleared or progresses towards aggregation. More recent work has also strengthened the link between amyloid and tau. For example, amyloid beta 42 enhances tau phase separation and may accelerate tau aggregation and fibril formation. So, blood-based tau biomarkers have become increasingly important in Alzheimer's disease as they offer a minimally invasive and scalable way to detect tau pathology. So among these, plasma phospho-tau 217 has emerged as one of the most robust and reliable markers of AD pathology. So multiple studies, very recent studies, have now shown that phospho-tau 217 levels strongly correlate with both amyloid and tau PET imaging, so meaning it reflects the underlying brain pathology quite accurately. It also tracks well with disease severity and progression. It's making it not just useful for diagnosis, but also for monitoring the progression of disease over time. So a major milestone came in 2025 with the FDA approval and clearance of the Lumipulse G phospho-tau 217 to beta amyloid 1:42 plasma ratio test. So this is one of the very first blood-based assays which have been approved to assess Alzheimer's-related amyloid pathology in symptomatic individuals. And it marks a significant shift towards clinical implementation. And so all of these advances represent a shift in the field so from relying on expensive and invasive methods like PET imaging and CSF sampling to more accessible blood-based diagnostics that could ultimately enable earlier disease detection and broader screening. And finally, therapeutic approaches. So these have been touched on by Irasima. But for focusing on just on tauopathies, they're become increasingly shifting towards targeting the underlying biology rather than just treating the symptoms alone. So there's several strategies in the tau field. So one of the major strategies involves antisense oligonucleotides. So these aim to reduce tau expression at the mRNA level. Also, monoclonal antibodies to target extracellular tau and also potentially block its spread between neurons. And small molecules are increasingly being explored to inhibit aggregation and modify tau function. So there is an increasing interest to combine all of these therapies in a kind of combinatorial fashion, let's say focusing on not just tau, but also increasing or integrating with treatments for amyloid. This is particularly apparent in obviously Alzheimer's disease, where two of these two proteins and pathologies interact. Overall, the field is moving towards a more personalized approach where therapies are matched to specific tau mechanisms, as well as the different types of and stage and subtype of disease. And so um, I use proteomics and lots of different omics tools and technologies in my research. So I just wanted to highlight um, some of the most recent advances in kind of omics and mass spectrometry and proteomics based tools in neurodegenerative disease research as a whole, not just tau biology. So nowadays, using the latest mass spectrometers and proteomics tools, we are able to look at ultra deep and towards single cell and spatial resolution of, let's say, brain tissues in Alzheimer's disease research. So this allows the comparison of different cell types and region specific proteome mapping. We are able to resolve and characterize different proteoforms of um, pathogenic proteins, such as tau, but also alpha-synuclein and TDP43. So we can capture these different isoforms. As I said, we can nicely compare the different PTM signatures across the proteins in disease and also proteolytic fragments. I use a lot of structural proteomics in my research. So there's quite a lot of advances in native and cross-linking mass spectrometry and hydrogen deuterium exchange mass spectrometry. And these are constantly being improved upon and integrated with more traditional structural tools such as X-ray crystallography and also, of course, cryo-EM. And these can help us link different protein conformations to aggregation and toxicity. Targeted mass spectrometry is being used and advanced to measure, let's say, phospho-tau 217, but also different phosphorylation sites and also overall protein abundance. So we can use these in a range of different biofluids, not just plasma, but also CSF, and of course, we can apply this to brain tissue as well. So all of this allows us to look at a more systems level approach where we can generate protein interaction networks, and this will ultimately help us reveal disease-associated pathways. So, of course, I'm quite interested in proteomics, but nowadays people integrate all of this with, lipidomics, transcriptomics, for example, and this allows us to identify causal drivers and novel therapeutic targets, not just for tau, but also for amyloid, alpha-synuclein, etc. So a lot of these would be very useful in your research. So that's the last slide I have. So I just want to say we're always looking for very talented people to come join us in Oxford. So if anyone's interested or with the presentation and some of the work I've just presented, please feel free to get in touch. And the final thing I want to say is, as part of the Trasna Nadanta initiative, for those of you who are attending the Alzheimer's Association International Conference in July in London, we're having a special networking event on the Monday evening of the conference. So this is at a pub very close to the Excel Centre where the conference is being hosted. So if you're free and want to connect with various different dementia researchers across Ireland and also the Irish diaspora, you're more than welcome to join us at that. So look out for further advertisement for that. And that's it. Thank you.